This lab is going to focus on photosynthesis and cellular respiration in algal beads. Your objectives for this lab are to observe photosynthesis and cellular respiration in algal beads in both the light and the dark. You're then going to design a lab investigation that analyzes factors that affect photosynthesis, such as light intensity, light color, or temperature. And then you're going to communicate your data in a scientific lab report. During experiment 1, when you observe the algal beads in both the light and the dark, you're going to be using this indicator sheet to determine the color of the carbon dioxide indicator in each of your cuvettes. During this experiment, you also want to label all of your pipettes, all of your cuvettes, and all of the beakers according to the protocol, and you want to make sure that you read all instructions. What I'm going to do is go through the experiment simplified, and then I'm going to go through it specifically step by step to help you through this lab. The first step is going to be to transfer the algal beads right from their little home in a mini beaker on your desk. You're then going to wash the algal beads with distilled water. You're going to add carbon dioxide indicator to the algal beads, and then you're going to set them in the light and the dark. And you're going to collect the data every 5 minutes into the shared Google Sheet, which you can find in the HyperDoc. Right here you can see a cuvette and a cap. The CO2 indicator is inside the cuvette, and the algal beads are sitting in the bottom. So this is just like a little mini home for them. It's where you're going to collect your data. What you're going to do with your partner is first label one cuvette light and the other cuvette dark. You can use a little L and a little D. You want to label it at the top where it says label here so it doesn't obstruct light reaching the algal beads. Then you're going to label transfer pipettes algae, and you're going to convert it into a scoop pipette by cutting it on an angle. Okay, and then you're going to use the algae pipette to transfer 10 algae beads into each of the light and dark cuvettes. You're going to label another pipette excess, and you're going to use it to remove and discard the liquid that might have gotten transferred along with the beads. You want to make sure not to grab the beads or try not to disturb them. Kind of move them to the side and insert the pipette into it to suck up that extra liquid. You're going to discard it into your labeled waste container beaker. You're going to label a new transfer pipette wash, and you're going to use it to add 1 ml of distilled water to each of the cuvettes. And you're going to let these algal beads incubate in the water for 5 minutes. So make sure that you time them for 5 minutes and let them incubate at room temperature. Now you're going to use that wash transfer pipette to remove that water that you just incubated the algal beads in, and you're going to put that water into the waste container. You're going to label another transfer pipette indicator for carbon dioxide, and you're going to transfer 1 milliliter of carbon dioxide into each cuvette, and then you're going to cap them tightly. You're going to wrap the cuvette that's labeled dark in aluminum foil, and you're going to place both the cuvettes labeled light and dark on their sides. You want to lay them flat so that the algal beads can kind of spread out like it shows you in this picture. The beads should be evenly distributed about the cuvette, so just kind of like shimmy it a little. And make sure that the clear side of the cuvette, you'll see when you get it, there's like a darker side and a clear side. The clear side should be facing the light. The cuvette should also be 15 to 25 centimeters from the light. So you want to make sure that you measure that. So you need to have this experimental setup double checked by me before you begin to collect your data. You're going to collect starting at time 0, check the color of the indicator, and then every 5 minutes you're going to mix the indicator by kind of gently rocking the cuvette back and forth, and then you're going to compare it to the color guide. We're not going to be using a spectrophotometer. Once you take these measurements, you want to be quick about them. One of you take the light, one of you take the dark, and you want to return them back to their experimental area as quickly as you can. You don't want to keep them out of the light too long. When you're collecting your data, there's a shared Google Sheet. You're going to go into that shared Google Sheet, add a new sheet at the bottom of the page with you and your partner's initials, and collect your data for experiment 1 here. You start with time 0. You want to look at the color and tell me what the pH is for each of these all the way up until 45 minutes. You're going to be using the information and the protocol that you use in this experiment to help you design your experiment for the next lab day. Good luck!